Hey there, my friends. Let's begin tonight with something quite extraordinary. Currently, as you're settling into comfort, as your breathing slows and your muscles begin to release the day's tension, something remarkable is happening inside you. Trillions upon trillions of tiny events are unfolding in perfect coordination. Molecules are dancing, instructions are being read, and an entire universe of activity hums along beneath your awareness. We're going to explore that hidden universe tonight. We're going to talk about DNA, cells, and the fundamental architecture that makes you possible. And we're going to do it slowly, carefully, the way one might describe a magnificent painting or a beloved piece of music. Now, settle in, dim the lights if possible, and let's drift into the fascinating yet often overlooked aspects of human anatomy. Think of your body as a city, not just any city, but one of those sprawling metropolises that stretches to the horizon, filled with millions of inhabitants, each with their own role to play. Except your body isn't home to millions. It's home to approximately 37 trillion cells. That number is so large it becomes almost meaningless, doesn't it? 37 trillion. If you tried to count them, one per second, it would take you over 1 million years. And each of these cells is not just a brick in a wall, but a living entity with its own complex machinery, its own purpose, and its own story to tell. Now, we often hear that cells are the basic units of life, and that's true enough. But what does that really mean? Imagine for a moment that you could shrink yourself down, smaller and smaller, past what the naked eye can see, past what even a magnifying glass could reveal, down to a scale where a single cell becomes as large as a building. What would you find there? You discover something that looks less like a simple blob and more like a bustling factory, a place of constant activity and purpose. The cell membrane would rise up around you like walls, not solid but flexible, studded with doorways and gates that open and close, allowing certain things in and keeping others out. This membrane is incredibly selective, remarkably intelligent in its own chemical way. Step inside this cellular factory and you'd find yourself in a space filled with a gel-like substance called cytoplasm. It's not quite liquid, not quite solid, but something in between. Floating in this cytoplasm are structures called organelles, each with its own specific job. There are mitochondria, those famous powerhouses of the cell, which take the food you eat and the oxygen you breathe and convert them into usable energy. There's the endoplasmic reticulum, a vast network of folded membranes where proteins are assembled and processed. There are Golgi bodies, which package proteins like a shipping department, preparing them for transport to wherever they're needed. And scattered throughout are ribosomes, tiny protein-making machines that we'll return to shortly because they're absolutely central to our story. But at the heart of this cellular world, enclosed in its own double-layered membrane, sits the nucleus. This is the command center, the library, the keeper of secrets. This is where the DNA lives. DNA. 3 letters that have become part of our cultural vocabulary, shorthand for identity itself. We talk about things being in our DNA when we mean they're fundamental to who we are. And in a very literal sense, that's exactly right. DNA, deoxyribonucleic acid, if we want to be formal about it, is the instruction manual for building and running a human being. But calling it an instruction manual doesn't quite capture the elegance of what it is. It's more like a library containing every book ever written about you before you were even born. The structure of DNA is famous now, that twisted ladder shape known as a double helix. 2 long strands wind around each other in a gentle spiral, connected by rungs that bridge the gap between them. It's a shape that James Watson and Rosalind Franklin and Maurice Wilkins and Francis Crick revealed to the world in the 1950s, though the story of that discovery is a human tale of brilliance and tragedy, and, it must be said, some injustice that we're still reckoning with today. But let's focus on the molecule itself, because it's genuinely beautiful. Each strand of the DNA ladder is made up of smaller units called nucleotides. Think of nucleotides as letters in an alphabet, except this alphabet has only 4 letters. We call them A, T, G, and C, which stand for adenine, thymine, guanine, and cytosine. These are the bases, the chemical compounds that form the rungs of that twisted ladder. Here's where it gets interesting. The bases don't pair up randomly. A always pairs with T. G always pairs with C. Always. This specificity, this chemical preference, is what makes DNA so incredibly stable and yet so perfectly suited for copying itself. Imagine writing a message where every letter is paired with a specific partner. If you know one strand, you automatically know the other. This is exactly how DNA works. When a cell needs to divide, when it needs to make a copy of itself, it doesn't have to somehow duplicate the entire twisted structure at once. Instead, the 2 strands separate, like a zipper being pulled apart, and each strand serves as a template for building a new partner strand. The result? 2 identical copies of the original DNA molecule, one for each new cell. It's elegantly simple, and yet the precision required to do this without errors is staggering. Now here's something to consider as you're relaxing into sleep tonight. The DNA in just one of your cells, if you could stretch it out into a straight line, would be about 2 meters long, 6 feet. And remember, you have 37 trillion cells. If you could take all the DNA from all your cells and lay it end to end, it would stretch from here to the sun and back more than 300 times. All of that information, all of that molecular instruction is coiled and packed and organized inside you right now. The compression algorithm required to fit 6 feet of DNA into a cell nucleus that's only about 6 micrometers across is something that would make any computer engineer weep with envy. But having all this information stored away doesn't mean much, unless there's a way to read it, to access it, to use it. And this is where things get truly fascinating. The way your cells use DNA is not at all like the way you might use a cookbook, reading straight through from beginning to end. Instead, your cells access DNA more like you might use an encyclopedia, looking up specific entries when they need specific information. And what are they looking up? Instructions for making proteins. Proteins are the workhorses of your body. They're structural elements, like the collagen that gives your skin its strength. They're enzymes that speed up chemical reactions, making possible in milliseconds what would otherwise take years. They're signaling molecules that carry messages from one part of your body to another. They're transporters, defenders, builders, and demolition crews. Everything your body does, every function it performs, involves proteins in some fundamental way, and every protein is built according to instructions encoded in your DNA. The segments of DNA that contain instructions for making proteins are called genes. You have somewhere around 20,000 genes, which might sound like a lot until you realize that a grape has more. A water flea has more. It turns out that complexity doesn't necessarily correlate with gene count. What matters more is how those genes are used, how they're regulated, and how they interact with each other. It's not just about having a large vocabulary, it's about how you combine words into sentences, sentences into paragraphs, and paragraphs into stories. The process of going from DNA to protein is one of the most fundamental processes in all of biology, and it happens in 2 main steps that scientists call transcription and translation. These are perhaps not the most inspired names, but they're actually quite appropriate. Let's walk through this slowly, because it's worth understanding. Transcription is the first step. When a cell needs to make a particular protein, it needs to access the gene that contains the instructions for that protein. But here's the thing. DNA is too precious, too important to let just wander out of the nucleus. It's like a rare manuscript that never leaves the library. So instead of bringing the DNA to where the proteins are made, the cell makes a copy of just the relevant section. This copy is made of a slightly different molecule called RNA, ribonucleic acid. Think of RNA as a working draft, a photocopy that can be taken out into the world and used. The process starts when proteins called transcription factors recognize the beginning of a gene and signal that it's time to make a copy. An enzyme called RNA polymerase arrives on the scene. This is a molecular machine of considerable sophistication. It latches onto the DNA at the start of the gene and begins to move along the strand, reading the sequence of bases. As it reads, it builds a complementary strand of RNA. Where there's a G in the DNA, it adds a C to the RNA. Where there's a C in the DNA, it adds a G. Where there's an A, it adds a U. In RNA, the base uracil takes the place of thymine, and on it goes, letter by letter, copying out the gene's message until it reaches a signal that says, "Stop here." The result is a strand of messenger RNA, or mRNA for short, carrying the protein-building instructions out of the nucleus. This mRNA molecule is then processed and edited in ways that we're still learning about. Sections that don't code for protein, called introns, are snipped out. The remaining pieces, called exons, are spliced back together. Sometimes they're spliced in different ways, which means a single gene can actually produce several different proteins depending on how the editing is done. It's economical, really. Why store the same information multiple times when you can cut and paste it in different combinations? Once the mRNA is ready, it travels out through pores in the nuclear membrane and into the cytoplasm. This is where translation happens, where the message carried by the mRNA is translated into the language of proteins. And this is where those ribosomes we mentioned earlier come into play. Ribosomes are themselves made of RNA and protein, assembled into two subunits that come together when it's time to build a protein. Think of them as reading machines, molecular devices that can scan along an mRNA strand and decode its message 3 letters at a time. Each 3-letter sequence, called a codon, specifies a particular amino acid. Amino acids are the building blocks of proteins, small molecules that can be linked together in chains. There are 20 different amino acids that your cells commonly use, and the order in which they're strung together determines what kind of protein you'll end up with. But how does the ribosome actually bring the right amino acid to the right place? This is where another type of RNA enters the picture, called transfer RNA or tRNA. Each tRNA molecule is specialized. On one end, it carries a specific amino acid. On the other end, it has a 3-letter sequence that matches a specific codon on the mRNA. The tRNA molecules float around in the cytoplasm, each carrying its designated amino acid, until they find their matching codon on an mRNA that's being read by a ribosome. When a match is made, the amino acid is added to the growing protein chain, and the tRNA floats away to pick up another amino acid and do it all again. The ribosome moves along the mRNA, codon by codon, and the protein chain grows longer and longer. When the ribosome reaches a codon that signals stop, the newly made protein is released. But it's not done yet. Proteins don't usually work as simple chains. They fold up into complex 3-dimensional shapes, and this shape is absolutely critical to their function. A protein's shape determines what it can do, what it can bind to, and what reactions it can catalyze. The folding happens partly on its own. The chemical properties of the amino acids guide the chain into certain configurations, but it's also assisted by other proteins called chaperones, which help ensure everything folds correctly. This entire process, from DNA to RNA to protein, is happening constantly in nearly every cell in your body, right now. As you breathe, as your heart beats, as your neurons fire and your muscles relax. It's happening billions upon billions of times per second. Each cell is reading its DNA, making copies, building proteins, and using those proteins to perform whatever specialized function that cell is designed for. And here's something worth pondering as you drift towards sleep. Every cell in your body contains the same DNA. The cells in your heart have the same genetic information as the cells in your brain, which have the same information as the cells in your liver or your bones or your skin. And yet they're all so different. A neuron looks nothing like a muscle cell. A red blood cell has a completely different job than a skin cell. How is this possible if they're all working from the same instruction manual? The answer is gene regulation, and it's one of the most elegant aspects of cellular biology. Just because a cell has a gene doesn't mean that gene is turned on. In fact, most genes in most cells are turned off most of the time. A neuron has all the genes necessary to make the proteins found in muscle cells, but those genes are silenced, kept quiet, and packed away in a form that can't be accessed. Meanwhile, the genes for neuron-specific proteins are active, being transcribed and translated. Each cell type has its own pattern of gene expression, its own set of genes that are switched on or off. And this pattern is what makes a heart cell and not something else. How does this switching work? There are proteins called transcription factors that can bind to DNA near genes and either promote or prevent transcription. There are chemical modifications that can be added to DNA or to the histone proteins that DNA wraps around, like tags that say, "Keep this gene closed," or "This one's okay to open." There are small RNA molecules that can interfere with translation, preventing certain mRNAs from being used even after they're made. The whole system is a marvel of regulatory control, layers upon layers of on-off switches and volume knobs and fine-tuning mechanisms. This regulation is what allows a single fertilized egg to develop into an entire human being. That first cell contains all the DNA you'll ever have. As it divides, the DNA is copied perfectly each time, so every descendant cell has the complete set of instructions. But as development proceeds, cells begin to specialize. Some become part of the nervous system. Others become muscle or bone or blood. Each lineage turns on certain genes and turns off others, gradually committing to a particular identity. It's a process of progressive specialization, and once a cell has committed to a particular fate, it generally sticks with it for life. Let's zoom out now, from the molecular scale back up to something we can more easily visualize. We've been talking about what happens inside cells, but we should also talk about the cells themselves, because they come in remarkable variety. Your body contains roughly 200 different cell types, each adapted for a specific function. Consider the neuron, the fundamental unit of your nervous system. Neurons are shaped like no other cell, long and branching, with a cell body that houses the nucleus and then an extended arm called an axon that can stretch for remarkable distances. Some neurons in your legs have axons that run all the way from your spine to your toes, making them among the longest cells in your body. Neurons communicate with each other through electrical and chemical signals, forming networks of stunning complexity. The human brain alone contains roughly 86 billion neurons, each one potentially connected to thousands of others. The number of possible connections is astronomical, greater than the number of stars in the observable universe. Or consider the cells of your immune system. White blood cells patrol your body constantly, searching for invaders. Some of them can actually change shape, squeezing through the walls of blood vessels to reach sites of infection. Others produce antibodies, Y-shaped proteins that can recognize and bind to specific foreign molecules. The immune system has a kind of memory built into it at the cellular level. Once your immune cells have encountered a particular pathogen, Some of them stick around as memory cells, ready to respond more quickly if that invader ever shows up again. This is how vaccines work, training your immune cells to recognize a threat before you ever encounter the real thing. Red blood cells are among the most specialized cells in your body and also among the strangest. In mammals, mature red blood cells don't have nuclei. They've ejected their DNA entirely, making more room for hemoglobin, the protein that carries oxygen. This means red blood cells can't divide, can't repair themselves, and can't make new proteins. They're essentially biological machines with a limited lifespan, about 4 months, after which they're broken down and recycled. Your body produces about 2 million new red blood cells every second to replace the ones that are wearing out. 2 million per second. Think about that. Right now, as you're resting, your bone marrow is churning out red blood cells at a rate that would make any factory jealous. Muscle cells are another fascinating case. They're packed with proteins that can slide past each other, creating the mechanical force of contraction. When a nerve signal reaches a muscle cell, it triggers a cascade of events that causes these proteins to grip each other and pull, and the cell shortens. When billions of muscle cells do this in coordination, you move. You walk, you breathe, and your heart beats. Cardiac muscle cells have a special feature. They're electrically connected to each other, allowing the contraction signal to spread rapidly across the entire heart. This is why your heart beats as a unified whole, rather than as a collection of individual cells twitching randomly. Epithelial cells form the linings of your body, covering your skin and the surfaces of your internal organs. These cells are tightly packed together, forming barriers that protect what's inside. The cells lining your intestines have special features on their surfaces, tiny finger-like projections that increase surface area for absorbing nutrients. The cells lining your lungs are extremely thin, allowing oxygen and carbon dioxide to pass through easily. And the cells on the surface of your skin are actually dead, dry husks filled with a tough protein called keratin. They form a protective layer that gets constantly worn away and replaced from below. You shed millions of skin cells every day, leaving a little trail of yourself wherever you go. All these different cell types, working together and performing specialized functions, form the tissues and organs of your body. And yet they all started from that single cell, that fertilized egg with its complete set of DNA. Development is perhaps the most remarkable journey in all of biology, the process by which one cell becomes many, becomes specialized, and becomes organized into a functional whole. Let's think about this development for a moment. After fertilization, the egg begins to divide. One cell becomes 2, 2 become 4, and 4 become 8. For the first few divisions, all the cells look pretty much the same, but then something interesting happens. The ball of cells begins to organize itself. An outer layer forms, an inner layer forms, and a space appears in the middle. This is the beginning of structure, the first hint that there's a plan at work. As development continues, the embryo forms 3 primary layers: the ectoderm, which will give rise to skin and the nervous system; the mesoderm, which will become muscle, bone, and blood; and the endoderm, which will form the lining of the digestive system and associated organs. From these 3 simple layers, everything else emerges. Cells migrate to new locations. They signal to each other, releasing chemicals that influence what their neighbors will become. Some cells even undergo programmed death, carefully controlled suicide that's necessary for proper development. The webbing between your fingers, for example, was removed through this process before you were born. The cells that formed that webbing received signals telling them to die. And they did, leaving behind separate digits. The signals that guide development are often proteins encoded by genes called developmental control genes. Some of these genes are ancient, conserved across hundreds of millions of years of evolution. The genes that help establish the basic body plan in a human embryo are similar to genes that do the same thing in fruit flies. This tells us something profound. The fundamental mechanisms of building a body are old, tested, and refined over eons. One of the most important principles in development is that cells don't just respond to chemical signals, they also respond to mechanical forces, to the physical pushes and pulls of their environment. A developing bone experiences mechanical stress as muscles begin to form and pull on it, and this stress actually influences how the bone grows and shapes itself. Even the way cells are packed together, the physical contact between them affects their behavior and identity. Life is not just chemistry, it's also physics, mechanics, and architecture. Throughout development and throughout life, cells maintain their identities through epigenetic mechanisms, modifications to DNA and its associated proteins that don't change. The actual sequence of bases but do affect which genes can be accessed. Think of your DNA as a vast library. Epigenetic modifications are like putting some books in locked cases while leaving others out on display. The information in those locked books is still there, but it's not available for use. These epigenetic marks can be remarkably stable, passed down through cell divisions, helping to ensure that a liver cell's daughter cells remain liver cells. Interestingly, some epigenetic marks can even be influenced by environmental factors. The food you eat, the stress you experience, and the chemicals you're exposed to, all of these can potentially affect epigenetic patterns. There's evidence that some of these marks can even be passed from parent to child, a sort of molecular memory of experience. This is still a controversial and actively researched area, but it suggests that the relationship between genes and environment is more nuanced than we once thought. You're not just the product of your DNA, you're the product of how that DNA interacts with the world around it. Now, we should talk about what happens when things go wrong, because understanding the normal functioning of cells also helps us understand disease. Mutations, changes to the DNA sequence, happen more often than you might think. Every time DNA is copied, there's a chance for an error. Most of these errors are caught and corrected by cellular proofreading mechanisms, molecular spell checkers that scan the newly made DNA and fix mistakes. But occasionally, an error slips through. Most mutations have no effect at all, occurring in parts of DNA that don't code for anything important. Some mutations are actually beneficial, providing the raw material for evolution, but some mutations cause problems. Cancer, fundamentally, is a disease of cells losing their normal controls. It typically requires multiple mutations accumulating in the same cell over time. One mutation might damage a gene that normally tells cells when to stop dividing. Another might disable a gene that triggers cell death when something goes wrong. Another might help the cell evade the immune system. When enough of these mutations pile up, a cell can become cancerous, dividing uncontrollably, ignoring signals that tell it to stop, and eventually forming a tumor. Understanding cancer at the cellular and molecular level has revolutionized how we treat it, leading to therapies that target specific proteins produced by cancer cells, drugs that help the immune system recognize and attack tumors, and treatments tailored to the specific mutations present in an individual's cancer. Other diseases result from mutations in specific genes. Sickle cell anemia, for instance, is caused by a single change in the gene for hemoglobin. One wrong amino acid in the protein chain, and the hemoglobin molecules behave differently, causing red blood cells to deform into a sickle shape. Cystic fibrosis results from mutations in a gene that codes for a protein involved in moving salt across cell membranes. When that protein doesn't work properly, mucus builds up in the lungs and other organs. Huntington's disease is caused by an expansion of a repeated sequence in a particular gene, like a stutter in the DNA that gets worse over generations. But here's something remarkable. Even with these genetic diseases, even with the potential for things to go wrong, The vast majority of the time, everything works. Your cells read their DNA accurately. Proteins fold correctly. Signals are sent and received. Cell division produces healthy daughter cells. The machinery of life, for all its complexity, is remarkably robust. There are backup systems, redundancies, and repair mechanisms. Evolution has had billions of years to refine these processes, and it shows. Let's think about that evolutionary perspective for a moment, because it adds a dimension to our understanding of cells and DNA. Every cell in your body is the descendant of cells stretching back in an unbroken line for billions of years. The first cells appeared on Earth somewhere around 3.5 to 4 billion years ago. Simple single-celled organisms without nuclei, without most of the elaborate machinery we've been discussing. But even those first cells had DNA, or possibly RNA, in the very beginning, and they had the ability to copy that genetic material and pass it on to the next generation. Over time, cells became more complex. About 2 billion years ago, a momentous event occurred. One cell engulfed another, but instead of digesting it, the two began living together in a mutually beneficial arrangement. This is how mitochondria came to be. They were once independent bacteria, now permanent residents inside our cells, still carrying their own small circle of DNA, still dividing on their own schedule. Every mitochondrion in your body is a descendant of those ancient bacteria, a living fossil of a merger that happened so long ago it's hard to fathom. Later, single cells began to cooperate, forming colonies, then true multicellular organisms. This required new innovations, ways for cells to communicate with each other, ways to divide labour, and ways to maintain organisation. The evolution of complex multicellular life was one of the great transitions in the history of Earth, and we are beneficiaries of that transition. Your body is a community of cells that long ago learned to work together. The DNA in your cells tells this evolutionary story. Hidden in your genome are sequences that resemble viral DNA, remnants of ancient infections that became incorporated into the genomes of our ancestors. There are genes that we share with yeast, with plants, and with bacteria. There are stretches of DNA that don't seem to do much of anything, sometimes called junk DNA. Though that term is increasingly seen as unfair, much of this DNA may have regulatory functions we're only beginning to understand. Your genome is not a pristine instruction manual written from scratch. It's a palimpsest, a document that's been written on, erased, rewritten, added to, edited, and revised over countless generations. As you're lying here now, comfortable and warm, your cells are expressing genes that your ancestors expressed millions of years ago. The genes that make it possible for you to process oxygen are ancient, because oxygen processing is fundamental to complex life. The genes involved in building your body plan during development are ancient, conserved across vast stretches of evolutionary time. But you also have genes that are newer, that appeared more recently as primates evolved, as humans diverged from other apes. Your genome is a history book, and you're the latest edition. We should spend a moment on stem cells because they represent something special in the landscape of cellular biology. Most cells in your body are specialists, committed to a particular identity and function. But stem cells retain the ability to become multiple different cell types. Embryonic stem cells found in early development can become anything. They're pluripotent, meaning they have the potential to form any cell type in the body. As development proceeds and the body takes shape, most of these broadly capable cells differentiate and lose their potency. But some stem cells persist into adulthood, tucked away in specific niches. Your bone marrow contains stem cells that continually produce new blood cells. Your skin has stem cells that generate new skin cells to replace the ones you shed. Your intestines have them. Even your brain has a small population of neural stem cells, though their role is still being understood. These adult stem cells are more limited than embryonic ones. They can typically only produce the cell types of the tissue they're in, but they're crucial for maintenance and repair. When you cut your skin, stem cells spring into action, dividing and differentiating to heal the wound. When you lose blood, bone marrow stem cells ramp up production. The presence of these stem cells means your body has a renewable quality, a capacity for regeneration, albeit limited. We can't regrow a lost limb the way a salamander can, but we're not entirely static either. In recent years, scientists have discovered ways to reprogram adult cells, coaxing specialized cells back into a stem-like state. These induced pluripotent stem cells have opened up new possibilities for research and therapy. Imagine being able to take a skin cell from a patient, turn it into a stem cell, and then guide it to become a neuron or a heart cell or whatever is needed. We're still in the early stages of realizing this potential, but the fact that it's possible at all is remarkable. It tells us that cellular identity, for all its stability, is not absolutely fixed. The locks on those library books can be opened under the right circumstances. Let's consider for a moment the sheer scale of coordination required for your body to function. 37 trillion cells, remember, each one with its own internal complexity, its own set of processes. And yet they all work together. Cells in your liver are detoxifying compounds absorbed from your gut. Cells in your pancreas are secreting insulin in response to rising blood sugar. Cells in your kidneys are filtering your blood, producing urine. Cells in your immune system are patrolling for threats. And none of this requires your conscious attention. You don't have to think about making your heart beat, or digesting your food, or filtering your blood. It all happens automatically, orchestrated by cellular communication and genetic programs and feedback loops. The communication between cells happens in multiple ways. Some cells release hormones into the bloodstream, chemical messages that travel throughout the body and affect any cell with the right receptor. Other cells communicate with their immediate neighbors through direct contact, proteins on one cell's surface binding to proteins on another's. Nerve cells communicate across synapses, tiny gaps where chemical neurotransmitters ferry signals from one neuron to the next. All of this signaling is, at its base, a matter of proteins. Proteins on cell surfaces, proteins released into the environment, and proteins inside cells responding to signals. And all of these proteins are encoded in DNA. There's an elegance to how it all fits together. DNA holds information. That information is copied into RNA. RNA is decoded to build proteins. Proteins do the work of the cell, including regulating which genes are expressed, completing the loop. Information flows from DNA to RNA to protein, but there are also feedback mechanisms, ways that proteins can affect gene expression, ensuring that the system is responsive and adaptive. We're learning that this flow of information is even more complex than the simple DNA to RNA to protein pathway suggests. There are RNAs that never get translated into protein, but instead have regulatory functions. There are chemical modifications to RNA that affect how it's used. There are three-dimensional structures that DNA forms inside the nucleus, bringing distant parts of chromosomes into contact with each other, allowing genes that are far apart in sequence to influence each other. The genome is not just a linear string of letters, but a dynamic three-dimensional entity, constantly being reorganized and accessed in sophisticated ways. Every time we think we understand how cells work, we discover another layer of complexity. And yet, for all this complexity, the basic principles remain beautifully straightforward. Cells need instructions, and those instructions are encoded in DNA. Cells need to build things, and they build them by stringing together amino acids according to those instructions. Cells need to regulate themselves, and they do so through a network of signals and feedback loops. Everything else is elaboration, sophisticated detail, built on a simple foundation. As you settle deeper into rest tonight, as your conscious mind begins to drift, and your body relaxes fully, your cells continue their work. In your bone marrow, stem cells divide, producing the components of your blood. In your gut, epithelial cells absorb nutrients from your last meal, passing them into your bloodstream. In your brain, neurons fire in patterns that generate dreams, though you're not quite there yet. Your heart muscle cells contract in their endless rhythm. Your immune cells patrol. Your liver cells process. And in every single one of these cells, DNA is being read, RNA is being made, and proteins are being assembled. You are, in a very real sense, a process as much as a thing. You're a pattern of activity, sustained by the constant work of trillions of cellular agents acting on instructions written in a molecular code that's been refined over billions of years. The atoms that make up your body will all be replaced, recycled, and renewed over time. The molecules that form you are temporary visitors, but the pattern persists. The information in your DNA ensures continuity, each cell passing on its instructions when it divides, maintaining your identity even as the physical substrate constantly changes. There's something almost poetic about the relationship between stability and change in biological systems. Your DNA is remarkably stable, copied with high fidelity, protected and preserved. And yet it's also responsive, its expression patterns changing in response to signals, to stress, and to time. You are both the same person you were 20 years ago, genetically identical, and a completely different person. Every cell renewed, every protein replaced. This paradox of constancy and flux is fundamental to life. We've covered a lot of ground tonight, from the twisted ladder of DNA to the specialized cells of your organs, from the molecular machines that read genetic information to the evolutionary history written in your genome. We've explored how a single fertilized egg becomes a complex organism, and how cells maintain their identities, and how they communicate and coordinate. We've touched on what happens when things go wrong, and marveled at how often things go right. But perhaps the most important thing to take from all of this is a sense of wonder. Wonder at the complexity hidden inside you. Complexity that operates entirely outside your awareness. Wonder at the elegance of the solutions that evolution has discovered. The economy of using a simple 4-letter code to specify everything about an organism, the ingenuity of the ribosome's translation mechanism, and the sophistication of gene regulation. Wonder at the fact that you're not just made of cells and molecules. You are cells and molecules, acting in concert, creating something greater than the sum of their parts. You are 37 trillion cells organized into a thinking, feeling, breathing whole. You are information encoded in DNA, expressed through RNA, manifested as protein, and integrated into tissues and organs and systems. You are the inheritor of billions of years of evolutionary refinement, carrying in your genome the echoes of every ancestor who came before you, all the way back to the first cells in ancient seas. You are a temporary pattern sustained by constant renewal, a ship of Theseus being rebuilt plank by plank even as it sails on. And right now, as you drift towards sleep, all of this is continuing. Your cellular machinery doesn't rest. DNA is being copied in preparation for tomorrow's cell divisions. Damaged proteins are being broken down and recycled. Mutations that arose during the day are being repaired by molecular proofreaders. Your body is maintaining itself, healing itself, and preparing itself for another day of existence. This happens every night, usually without you giving it a single thought. But tonight, perhaps, you can appreciate it. You can feel grateful for the elegant molecular systems that make you possible. The instructions that guide all of this, the DNA in your cells, are not a rigid blueprint, but a flexible script, interpreted differently in different contexts, responsive to signals and circumstances. You are not simply the readout of your genetic code. You're the product of that code interacting with your environment, your experiences, and your choices. Nature and nurture are not separate influences but interwoven threads in the same tapestry. Your genes affect your behavior, and your behavior affects your gene expression. It's all one system, all connected, all part of the same story. So as you close your eyes tonight, as your breathing becomes slow and regular, as your mind lets go of the day's concerns, remember that you're part of something ancient and ongoing. The mechanisms that keep you alive are the same basic mechanisms that have kept life going on Earth for billions of years. You're a link in an unbroken chain stretching back to the beginning of life and forward to whatever comes next. The cells that make up your body are the descendants of the first cells, carrying forward the same fundamental processes, the same elegant solutions to the challenges of existence. Sleep well, knowing that your body knows what to do. Your cells will continue their work while you rest. DNA will be read, proteins will be made, energy will be generated, and repairs will be completed. When you wake, you'll be slightly different at the molecular level than you are now. Some cells will have divided, some proteins will have been replaced, some memories will have been consolidated, but you'll still be you, held together by the information in your DNA and the emergent properties of trillions of cells working in harmony. There's a rhythm to cellular life that mirrors, in some ways, the rhythms of your own existence. Cells are born when a parent cell divides. They mature, carrying out their specialized functions. Eventually, most of them die, either through programmed cell death when they're no longer needed, or simply through the wear and tear of existence. In your body right now, millions of cells are dying every second, and millions more are being born to replace them. This constant turnover, this endless cycle of death and renewal, is part of what keeps you healthy. Old cells with accumulated damage are removed. Fresh cells take their place. You are being continuously regenerated, though so gradually you never notice it happening. The lifespan of different cell types varies dramatically. Those red blood cells we mentioned earlier last about 4 months. The cells lining your stomach survive only a few days before they're sloughed off and replaced. They have to endure harsh acidic conditions, so they wear out quickly. Skin cells might last a few weeks. Some white blood cells live for years, maintaining immunological memory. And then there are neurons, most of which are with you for life, never dividing once they've matured. The neurons you're using to process these words right now, to imagine the cellular landscapes we've been describing, are likely the same neurons that were formed before you were born, refined and connected during early development, and maintained ever since. This brings up an interesting philosophical question. If most of your cells are replaced over time, are you still the same person? The answer, of course, is yes, but not because of physical continuity. You're the same person because of informational continuity. The pattern persists even as the material changes. Your DNA provides the template that ensures new cells resemble old ones. Your brain, with its relatively stable population of neurons, maintains memories and personality across time. You are, in essence, a pattern that has learned to perpetuate itself, an organization of matter that resists entropy through constant work and constant renewal. Let's think for a moment about the energy required for all this cellular activity. Every movement of a molecule, every chemical reaction, and every signal sent between cells requires energy. This energy ultimately comes from the sun, captured by plants through photosynthesis, stored in chemical bonds in the food you eat, and released by your cells through cellular respiration. Inside your mitochondria, organic molecules from your food are broken down in a controlled way, and the energy released is used to create ATP, adenosine triphosphate, the universal energy currency of cells. ATP is a beautifully simple molecule for such an important role. It's similar to the nucleotides that make up DNA and RNA. In fact, the A in ATP is the same adenine we talked about earlier. But ATP has 3 phosphate groups attached to it, and the bonds holding those phosphates contain energy. When a cell needs to do work, it breaks one of those bonds, converting ATP to ADP, adenosine diphosphate, and uses the released energy. Then, in the mitochondria, that ADP is recharged back to ATP, ready to be used again. This cycle happens constantly. You produce and consume roughly your own body weight in ATP every single day. Think about that. Every day your cells make and use about as much ATP as your entire body weighs. The turnover is staggering. The mitochondria that generate this ATP are, as we mentioned, descended from bacteria that took up residence inside cells billions of years ago. They still resemble bacteria in many ways. They have their own DNA, separate from the DNA in your cell nucleus. They have their own ribosomes. They divide independently of the cell they're in on their own schedule. And here's something interesting. Mitochondrial DNA is inherited exclusively from your mother. The egg cell contributes all the mitochondria to the fertilized egg. Sperm contribute essentially none. This means you can trace your maternal lineage back through mitochondrial DNA, a separate genetic record passed down from mother to daughter across generations, all the way back to a woman who lived in Africa roughly 150,000 years ago, the matrilineal ancestor of all living humans. This idea that your cells contain multiple genomes, the nuclear genome inherited from both parents and the mitochondrial genome inherited from your mother, is just one example of the complexity and layered history of cellular biology. Your cells are not simple units, but ecologies in their own right, communities of molecules and structures with different origins working together toward common goals. We should talk about the cell membrane in a bit more detail because it's more sophisticated than simply being a wall that keeps the inside in and the outside out. The membrane is made primarily of lipids, fat molecules arranged in a double layer. These lipids have a water-loving head and water-fearing tails, and they naturally arrange themselves so the heads face outward toward the watery environments on both sides of the membrane, while the tails cluster together in the middle, away from water. This creates a barrier that water-soluble molecules can't easily cross. But embedded in this lipid bilayer are proteins, thousands of different kinds, each with specific functions. Some proteins form channels that allow particular molecules to pass through. Others are pumps that actively transport molecules against their concentration gradient, using energy to move things from where they're less concentrated to where they're more concentrated. Still others are receptors that bind to signaling molecules outside the cell and trigger changes inside the cell in response. The membrane is not a static barrier, but a dynamic, selective interface, constantly monitoring the environment and controlling what enters and exits. Some molecules can pass through the membrane easily. Oxygen and carbon dioxide, small and uncharged, slip through without help. But larger molecules, or charged molecules like ions, need assistance. The sodium-potassium pump, one of the most important proteins in your cell membranes, constantly pumps sodium ions out of cells and potassium ions in, maintaining a concentration difference that's essential for nerve signaling, muscle contraction, and other processes. This pump uses a substantial fraction of your body's ATP. Maybe a third of all the energy you expend goes toward running sodium-potassium pumps. It's expensive energetically, but absolutely necessary. The way nerve cells use this ion gradient to generate electrical signals is particularly elegant. When a nerve is stimulated, channels in the membrane open, allowing sodium ions to rush in. This changes the electrical charge across the membrane, and this change itself triggers nearby channels to open, creating a wave of electrical activity that travels down the nerve fiber. It's self-propagating, like a row of falling dominoes. When the signal reaches the end of the nerve, It triggers the release of neurotransmitter molecules that diffuse across the synapse to the next cell. And then, after the signal has passed, pumps work to restore the original ion concentrations, resetting the system so it's ready to fire again. Your thoughts, your perceptions, and your movements, all of these depend on cells carefully managing the flow of ions across membranes. We've talked about how cells use DNA, but we should also acknowledge how much we still don't fully understand. For all our knowledge of genetics and cell biology, there are enormous gaps in our understanding. We know that most DNA doesn't code for proteins, but we're still discovering what all that non-coding DNA does. We know that the three-dimensional folding of proteins is critical to their function, but we can't yet reliably predict how a protein will fold based solely on its amino acid sequence. We know that cells communicate in complex ways, but we're still mapping out all the signaling pathways and feedback loops. We know that gene expression is regulated by multiple interacting factors, but we can't yet fully predict how a cell will respond to a particular combination of signals. Biology is messier than physics or chemistry. There are fewer universal laws and more context-dependent rules. Evolution is a tinkerer, not an engineer, building new solutions on top of old ones, repurposing existing structures for new functions, and creating complexity through accumulation rather than through elegant design. This means that biological systems often have redundancies, backup pathways, and multiple ways to accomplish the same thing. It also means there are quirks and inefficiencies, historical accidents frozen into our biology. The recurrent laryngeal nerve, for example, takes an absurdly long path from your brain to your larynx, looping down around major blood vessels in your chest before heading back up. In a giraffe, this nerve can be 15 feet long, making a ridiculous detour. Why? Because of evolutionary history. In fish, the ancestors of all land vertebrates, this routing made sense. As necks evolved and lengthened, the nerve came along for the ride, getting stretched but maintaining its original path. It's inefficient, but evolution doesn't optimize from scratch. It modifies what already exists. Your genome is full of similar historical artifacts. You have genes that were once functional but are now broken, pseudogenes that accumulate mutations because there's no selective pressure to maintain them. You have endogenous retroviruses, remnants of viral infections in your ancestors' germline cells, now passed down as part of your genetic heritage. Some of these viral sequences have even been co-opted for useful purposes. There's evidence that some proteins important for placental development originally came from viral genes. Evolution is opportunistic, using whatever materials are available, even if they came from parasites. This messiness, this historical contingency, is part of what makes biology so rich and interesting. You can't understand cells and DNA purely through first principles. You have to understand history, you have to understand evolution, and you have to appreciate that what exists is not necessarily optimal, but simply good enough to have survived. Life is not a perfect machine. It's a cobbled together collection of solutions that have managed, somehow, to persist and reproduce for billions of years. And yet, for all the messiness, for all the historical accidents and jury-rigged solutions, life works. Your cells function. Your body maintains itself. You exist, conscious and aware, pondering your own cellular basis. That's perhaps the most remarkable thing of all. Somehow, the complex interactions of molecules in your brain and the firing of neurons according to patterns influenced by genes, experience, and chance give rise to subjective experience. You're not just a collection of cells, you're a person who feels and thinks and dreams. The emergence of consciousness from cellular activity is one of the deepest mysteries in science, one we're only beginning to seriously investigate. Some researchers approach consciousness by studying the brain at the cellular level, mapping connections between neurons, and identifying which patterns of activity correlate with which experiences. Others look at the molecular level, studying how neurotransmitters and other signaling molecules affect mood and cognition. Still others take a more holistic view, looking at large-scale brain dynamics, the coordinated activity of billions of neurons working together. Probably the answer lies at all these levels simultaneously. Consciousness is not located in a single cell or a single molecule, but emerges from the organization and interaction of the whole system. Your neurons are talking to each other right now, even as you relax towards sleep. Patterns of activity wash across your brain, representing thoughts, memories, and the processing of these words. When you sleep, different patterns emerge: the slow waves of deep sleep and the rapid firing of REM sleep when dreams occur. These patterns are generated by cells, by the opening and closing of ion channels, by the release and uptake of neurotransmitters, and by the complex network of connections that link neurons together. Consciousness is grounded in cellular biology, even if we don't yet fully understand how the grounding works. There's something humbling about realizing how much of what you are operates below the level of your awareness. You don't consciously control your cells. You don't decide when to transcribe a gene or where to send a particular protein. You don't manage your ion gradients or coordinate your cellular communication. All of this happens automatically, the result of billions of years of evolutionary refinement. In a sense, you're a passenger in your own body, benefiting from cellular machinery you didn't design and couldn't consciously operate even if you wanted to. And yet, you're not entirely separate from this cellular activity. Your choices affect your cells. The food you eat provides the raw materials they need. The exercise you do, or don't do, influences how they develop and function. The stress you experience changes patterns of gene expression. The sleep you're settling into now gives your cells time to perform maintenance tasks that are difficult to accomplish while you're active. There's a feedback loop between you and your biology. You are your cells, and your cells are influenced by how you live your life. This intimate connection between the molecular and the experiential, between the cellular and the personal, is worth contemplating. When you feel happy, that feeling has a molecular basis. Neurotransmitters like dopamine and serotonin binding to receptors on neurons, triggering signaling cascades that alter cellular behavior and somehow create the subjective experience of happiness. When you remember something, that memory is encoded in the pattern of connections between neurons, in synapses that were strengthened through molecular changes triggered by experience. When you learn something new, genes are expressed in your brain cells, proteins are made, and your neural network physically reorganizes itself. Every thought, every feeling, and every experience leaves a trace in your cellular biology. This doesn't diminish the reality of your experiences. Knowing that love involves oxytocin and vasopressin doesn't make love less real or less meaningful. Knowing that memory involves protein synthesis and synaptic strengthening doesn't make your memories less precious. Understanding the mechanism doesn't erase the phenomenon. If anything, it makes it more wondrous. The fact that subjective experience can emerge from the interactions of molecules, that consciousness can arise from cellular activity, that you can exist as both a physical system and an experiencing subject. This is profound and beautiful, and deeply mysterious. As you lie here now, your body is preparing for sleep in ways both conscious and unconscious. Your conscious mind is letting go, thoughts becoming less directed and more dreamlike. But your cells are also preparing. Melatonin, a hormone released by your pineal gland in response to darkness, is binding to receptors throughout your body, signaling that it's time to rest. This affects gene expression in various tissues, adjusting metabolism for the sleep period. Your core body temperature is dropping slightly, part of the natural circadian rhythm that helps regulate sleep. In your brain, the balance of neurotransmitters is shifting, with wake-promoting chemicals declining and sleep-promoting ones increasing. During sleep, your cells will perform important maintenance work. Growth hormone will be released, stimulating cell division and protein synthesis, helping to repair tissues damaged during the day. In your brain, the glymphatic system, a sort of waste removal system, will become more active, clearing out metabolic byproducts that accumulated while you were awake. Some researchers believe this cleaning process is one of the key functions of sleep, that your brain needs downtime to flush out cellular debris that would otherwise build up and cause problems. Throughout your body, cells will be dividing, differentiating, dying, and being born. The constant renewal continues night and day. Your immune system too is affected by sleep. Certain immune cells are more active during sleep, patrolling more vigorously for pathogens. Inflammatory signaling molecules called cytokines, which help coordinate immune responses, are produced in different patterns during sleep than during wakefulness. This is part of why sleep is so important for fighting off infections. Your immune cells do some of their best work while you rest. Conversely, chronic sleep deprivation can impair immune function, making you more susceptible to illness. The connection between sleep and cellular health runs deep. Even your DNA repair processes are affected by circadian rhythms. There's evidence that the cellular machinery that fixes damaged DNA operates more efficiently at certain times of day. This makes sense from an evolutionary perspective. DNA damage is often caused by ultraviolet radiation and oxidative stress, both of which are higher during waking hours when you're active. Scheduling DNA repair for nighttime, when you're less active and generating fewer reactive oxygen molecules, is an efficient strategy. All of these processes, from hormone release to immune activity to DNA repair, are coordinated by your circadian clock, an intrinsic timekeeping system that exists in nearly every cell in your body. The master clock sits in a small region of your brain called the suprachiasmatic nucleus, a cluster of about 20,000 neurons that maintain a roughly 24-hour rhythm even in the absence of external cues. This master clock is synchronized to the day-night cycle by light-sensitive cells in your eyes, and it in turn synchronizes peripheral clocks in other tissues through hormonal and neural signals. At the cellular level, the circadian clock involves a feedback loop of gene expression. Certain genes are transcribed and translated to produce proteins that, after a delay, inhibit their own transcription. As these proteins degrade over time, the inhibition is lifted and the cycle begins again. This molecular oscillation, refined and regulated by many additional factors, creates a rhythm that influences everything from metabolism to cell division to cognitive performance. You are, in a very literal sense, a ticking clock with rhythms built into your cellular biology. These circadian rhythms are ancient. Even single-celled organisms like cyanobacteria have internal clocks that regulate their daily activities. The molecular mechanisms are different in different organisms, but the basic principle, using feedback loops of gene expression to create an oscillation, is remarkably conserved. This suggests that having an internal sense of time and anticipation of daily cycles provides such a strong survival advantage that it evolved early and has been maintained ever since. Your cells know what time it is, even without external cues, because that knowledge is written into their genes. As you sink deeper into rest, as the boundaries between waking and sleeping blur, your cellular activity continues unabated. Ribosomes translate mRNA into protein. Mitochondria generate ATP. Ion pumps maintain gradients across membranes. DNA is replicated in cells preparing to divide. Damaged proteins are tagged for degradation and broken down into amino acids that will be recycled into new proteins. The molecular machinery that sustains you never sleeps, never pauses, and continues its work from the moment you're conceived until the moment you die. And when death comes, eventually, to all of us, it too is a cellular phenomenon. Some cells die through necrosis, essentially being destroyed by injury or lack of oxygen, bursting open and spilling their contents. But many cells die through apoptosis, programmed cell death, an active process where the cell essentially takes itself apart in an orderly fashion. Apoptosis is not a failure. It's a normal, healthy part of life. It's how your fingers separated during development. It's how your immune system eliminates cells that might become cancerous. It's how old cells make way for new ones. A cell undergoing apoptosis shrinks. Its DNA is cut into fragments. Its membrane forms blebs, little bubbles that pinch off. The cell contents are packaged into vesicles that can be cleaned up by other cells without causing inflammation. It's a remarkably tidy process. Death without mess or fuss. When you die, your cells will gradually cease functioning, but many of them will activate their apoptosis programs in the process, dying cleanly rather than simply falling apart. This might sound morbid, but there's something almost comforting about it too. Death is not separate from life, but part of the same cellular processes that sustain you. The machinery that allows for programmed cell death is the same machinery that allows for growth and renewal. You cannot have one without the other. Life and death are intertwined at the most fundamental level, two aspects of the same biological reality. But let's return to life, to the here and now, to your cells working tirelessly to maintain you. There's so much happening that we haven't even touched on. We haven't talked about the endoplasmic reticulum in detail, that vast network of membrane-bound channels where proteins are folded and modified. We haven't discussed the Golgi apparatus, with its stacks of membrane compartments where proteins are further processed and packaged for transport. We haven't explored lysosomes, the cellular recycling centers where old organelles and proteins are broken down. We haven't delved into the cytoskeleton, the network of protein filaments that gives cells their shape and allows them to move and divide. Each of these cellular components could be the subject of an entire lecture. Each represents a marvel of molecular engineering, a solution to particular challenges faced by cells. The endoplasmic reticulum allows for protein folding in a controlled environment, separate from the cytoplasm. The Golgi apparatus acts as a sorting station, ensuring proteins get sent to the right destinations. Lysosomes allow cells to safely break down potentially dangerous materials in contained compartments. The cytoskeleton provides structural support while remaining dynamic and responsive, able to reorganize quickly when needed. And beyond the components we've mentioned, there are others we haven't discussed at all. Peroxisomes, which break down fatty acids and detoxify certain chemicals. Centrioles, which help organize cell division. The nuclear pore complexes are elaborate structures that control what moves in and out of the nucleus. The extracellular matrix is the network of proteins and carbohydrates that cells secrete to build the structural scaffolding of tissues. The list goes on and on. Cell biology is vast, intricate, and endlessly fascinating. Perhaps what's most remarkable is how much we've learned in a relatively short time. The cell was first observed in the 1600s when Robert Hooke looked at cork under a microscope and saw what he called cells, though what he was actually seeing were the empty cell walls of dead plant cells. The nucleus was identified in the 1830s. The idea that all organisms are made of cells was formalized in the 1830s and 1840s. The chromosome theory of inheritance emerged around 1900. DNA structure was solved in 1953. The genetic code was cracked in the 1960s. Restriction enzymes and recombinant DNA technology in the 1970s. The first human genome sequence was completed in 2003. CRISPR gene editing in the 2010s. In just a few centuries, we've gone from not knowing cells existed to being able to read and edit the genetic code. We can insert genes from one organism into another. We can use bacteria to produce human insulin for treating diabetes. We can modify immune cells to attack cancer. We can sequence an individual's entire genome in a matter of hours. The pace of discovery has been breathtaking, and it shows no signs of slowing down. Every year brings new insights into cellular biology, new understanding of how DNA works, and new appreciation for the complexity and elegance of the molecular systems that make life possible. And yet, for all this knowledge, when you look at a living cell under a microscope, there's still a sense of mystery, a quality of aliveness that's difficult to capture in diagrams and descriptions. A cell is more than the sum of its molecules. It's an integrated system, a dynamic whole that maintains itself, responds to its environment, and participates in the larger organism. There's an organizational principle at work, a coordination of parts that creates something qualitatively different from what you'd get if you just mixed all those molecules together in a test tube. This emergent quality of life, the way complexity arises from the interaction of simpler components, is what makes biology so captivating. You can understand every molecule in a cell and still not fully grasp how they all work together to create a living system. You can map every connection in a brain and still not fully explain consciousness. There are levels of organization, each with its own logic, and understanding one level doesn't automatically give you understanding of the next. But we keep trying, keep exploring, keep peeling back layers of complexity to reveal deeper patterns, and in doing so, we learn not just about cells and DNA, but about ourselves, about where we came from, and about what we are. We learn that we're part of nature, not separate from it. We learn that the same principles that govern the simplest bacteria also govern us. We learn that life is fundamentally unified, all connected through shared ancestry and shared molecular mechanisms. There's a perspective shift that happens when you really internalize what cellular biology teaches us. You realize that you're not a fixed, static thing, but a process, a flow of matter and energy maintained far from equilibrium. You're a wave propagating through space and time, sustained by the constant work of your cells. You're temporary, contingent, and improbable. The fact that you exist at all, that this particular arrangement of atoms organized itself into cells, tissues, organs, and eventually into a conscious being capable of contemplating its own existence, this is genuinely miraculous, not in a supernatural sense, but in the sense of being beyond what seems possible, and yet here you are. Your cells don't know they're part of you. They don't have awareness of the larger organism they constitute. They simply follow their programming, responding to local signals and carrying out their specialized functions. And yet, collectively, they create you. Consciousness emerges from unconscious parts. Intention emerges from molecular processes that have no intention. Meaning emerges from meaningless chemistry. These are the great paradoxes of biology, the mysteries that keep scientists and philosophers awake at night, even as your cells work to carry you into sleep. So rest now, knowing that you're in good hands, or rather, in the good molecular machinery of 37 trillion cells. They know what to do. They've been doing it for as long as you've been alive, and they'll continue until the end. You don't have to manage them, or worry about them, or even think about them. They operate on principles established long before you were born, refined through eons of evolution, and encoded in DNA that you inherited from your parents, who inherited it from their parents, back and back through time to ancestors who lived in trees, who lived in burrows, and who crawled in oceans, all the way to those first simple cells that appeared on a young Earth billions of years ago. You carry that history with you, written in your genes, expressed in your cells. You are a living link in a chain that stretches back to the very origin of life and forward to whatever future life may have. Your cells are nature's answer to entropy, to the tendency of organized things to break down. They maintain organization through constant work, building order from disorder, sustaining the improbable arrangement we call a living being. And with that thought, with gratitude for the hidden machinery that makes everything possible, and with appreciation for the molecular elegance underlying existence, let yourself drift completely now. Let your conscious mind release its grip. Your cells will continue their ancient work. The DNA will be read, the proteins will be made, and the energy will be generated. Life goes on at levels both far beneath and far beyond conscious awareness. You are held in a web of molecular processes, supported by cellular activities, and sustained by information encoded in the double helix. Sleep well. Rest deeply. Your cells are taking care of you.